I'm Andrea Musacchio and I'm a director at the Max Planck Institute of Molecular Physiology in Dortmund. The current focus of my activities is the study of the cell cycle. That is essentially a study of how cells divide. And we do this with a variety of approaches through, if you wish, an overarching approach called structural biology. We use structural biology, which means that we try to understand the molecular organization of certain structures within the cell that are responsible for dividing the chromosomes, so the genetic material, during cell division. So we studied them to understand what they look like molecularly and how they function. And so we try to combine techniques of reconstitution in vitro, so we try essentially to reproduce a cellular process in a test tube. We try to study the structures and we try to go back to the cells to understand how these processes that we have studied in vitro really impinge on the life of a cell. Well, Cryo-EM has revolutionized the way we do structural work and how we understand what molecules look like in 3 dimensions, which is essential to understand also how they operate, what kind of function they have. And Cryo-EM had a revolution literally in the last 10 years. First in the study of proteins in vitro, but more and more through techniques called tomography, cryo-electron tomography, also in cells. And so we can now understand the organization of macromolecular complexes in cells and derive functional hypotheses by imaging these large objects as they operate in their natural environment. So Cryo-EM helps essentially every aspect of my research. Well, the most exciting advancements in Cryo-EM technology at the moment are in the area of EM tomography, so essentially going into cells to image molecules in the natural environment, as I said earlier. But what I think will be transformative in the future will be the ability not only to image through cryo-EM tomography these molecules in their environment, but also to image them through fluorescence microscopy. So I believe that the real revolution will come when we will be able to image at super resolution, so beyond the limits of the resolution of light, so single molecules in our cells together with EM tomographic reconstruction, because this will give us the identity of molecules in cells, so we will be able to see where they are and from this derive hypotheses for how these molecules function.